Hello, good afternoon, everybody.
Good afternoon, sir.
Good afternoon, sir.
Okay, that means you are hearing me. Now we will start the class. Okay, can you see the slides?
Yes, sir.
Yes, sir.
Okay, then we'll begin. We're in respiratory system, and in our previous class we studied about the oxidation reduction reactions, and we gave importance to biological oxidation. What we said was we get energy from the food stocks we take, and how we get energy is due to the oxidation of food stocks. That is known as biological oxidation, or in simpler terms, cellular respiration, or even simpler internal respiration. And we also discussed about the different enzymes which were involved in oxidation reduction reactions. And of the same class of enzymes, the mechanism of action were different. Accordingly, we classified into oxidase, oxygenase, hydroperoxidase. But today we'll go to the TCA cycle. And before going to the TCA cycle, we'll discuss about the pyruvate dehydrogenase complex. Okay. What you know about the TCA cycle? This TCA cycle, is this the cycle for carbohydrate, protein, lipids, or what?
For all of them.
Okay, TCA cycle, tricarboxylic acid cycle, is the central metabolic pathway. About 65 to 70% of the energy in our body is found from the TCA cycle. Whether we metabolize carbohydrate, lipids, or protein, first of all, they will form the acetyl coenzyme A, and after forming the acetyl coenzyme A, they will go via the TCA cycle. That's why when saying about the TCA cycle, you should not forget to say that TCA cycle is the central metabolic pathway for all. It is carbohydrate, lipids, and proteins. Okay. But while studying in the class, we study like glycolysis, and glycolysis is followed by TCA cycle. Not only from the glycolysis, but from the metabolism of protein and lipids also, acetyl coenzyme is formed, and that goes to the TCA cycle. Okay. When we follow from the glycolysis, the glucose molecule is broken down into two molecules of pyruvate, and now that pyruvate will be converted to acetyl coenzyme A, and that acetyl CoA will go to the TCA cycle. That means the one reaction, that is the conversion of pyruvate to acetyl coenzyme A, is catalyzed by pyruvate dehydrogenase complex. First of all, we will study about this enzyme. Okay. In which cellular compartment, which part of the cell does glycolysis occur?
Cytoplasm.
Cytoplasm. Okay. And do you know about the TCA cycle? Where does it occur?
Mitochondria.
Okay, TCA cycle occurs in the mitochondrial matrix. Okay, simply mitochondria or the mitochondrial matrix. Okay, but this is the connecting reaction, that is the reaction of pyruvate dehydrogenase complex. That means the conversion of pyruvate to acetyl coenzyme. Where does that step occur? In cytosol or in mitochondria? Okay, that steps also occur in mitochondria, because after the formation of pyruvate, that pyruvate will be carried to the mitochondria, and in the mitochondria itself, that will be converted to acetyl coenzyme A and go through the TCA cycle. Okay. In today's class, first of all, we'll discuss about the pyruvate dehydrogenase, the reaction of the pyruvate dehydrogenase complex. And when saying about name of this enzyme, we usually say about complex. Why you say complex, and that also will go. And after that, we will start the TCA cycle. And about the regulation of TCA cycle and more, we'll deal in our next class. Okay. This is the general structure of a eukaryotic cell. Okay, in the cytosol, glucose is converted to pyruvate. Now this pyruvate will go to the mitochondria, and the TCA cycle will occur in the mitochondria. After that, electron transport chain, oxidative phosphorylation, all occurs in the mitochondria. Okay, this is just to show you the compartments of the cell. Glucose to pyruvate in the cytosol, and now this pyruvate will be carried to the mitochondria, and the further reactions will occur in the mitochondria. Okay, if you look the structure of mitochondria, you can see this is the double layered membrane. This is having the double layered membrane, and the outer membrane is the continuous, whereas the inner membrane is the folded. Inner membrane is folded in such a way that it gives rise to the cristae, and the inner side is known as matrix. Similar to the cytoplasm of the cell, now the mitochondria is having the matrix. This is this folded structure cristae. This you can see the intermembrane space. This intermembrane space you will have to deal with in detail while studying about the electron transport chain and oxidative phosphorylation. Okay, now this is the inner membrane, this is the outer membrane. Okay, this much, and this is the electron microscopic view of the mitochondria. Okay, biochemical anatomy of the mitochondria. Outer membrane and the inner membrane. We have already said, and in this there are certain porin channels. Okay, this outer membrane is freely permeable to small molecules and ions, whereas the inner membrane is not like that. Outer membrane is freely permeable to small molecules and ions, whereas the inner membrane, this is not that permeable. It is impermeable to most small molecules and ions, including hydrogen ions. And what does this inner membrane contain is the respiratory electron carriers from complex 1 to complex 4, that is different complex of electron transport chain. ADP ATP translocase, that means you all know that the mitochondria is the powerhouse of the cell. That means the energy is generated in the mitochondria, energy is generated in the form of ATP, and that ATP has to be taken out from the mitochondria, and the enzyme is ADP ATP translocase. For the synthesis, ATP synthase and other membrane transporters are there. Okay. ATP synthase, FO, F1, this portion is in the inside. Okay. From this slide, what you have to learn clearly is mitochondria is having outer membrane and inner membrane. You all know outer membrane is freely permeable to small molecules and ions, whereas the inner membrane is impermeable to most small molecules and ions, including hydrogen ion. And this inner membrane contains the respiratory electron carriers, that is the complex of electron transport chain, as well as the enzymes involved in oxidative phosphorylation and the enzyme translocating the formed ATP to the portion outside of the mitochondria. Now let's see the matrix. This mitochondrial matrix contains enzymes, pyruvate dehydrogenase complex, which we have just spoken, which converts pyruvate to acetyl coenzyme A, that is also in the mitochondrial matrix. Citric acid cycle enzymes, or the TCA cycle enzymes, are in the mitochondrial matrix. Beta oxidation, that means the oxidation of lipids, usually occur by the process of beta oxidation. That beta oxidation enzymes are also in the mitochondrial matrix, and amino acid oxidation enzymes are also here. In addition to that, in mitochondrial matrix, there is also DNA and ribosomes. You all know that DNA is present in the nucleus, but little amount of DNA is also present in the mitochondria, which is known as the mitochondrial DNA. Along with this, it also contains many other enzymes, ADP, ATP. Okay, no need to mention all these things. These are contained in the mitochondrial matrix. Now, to focus our class today, we have the pyruvate dehydrogenase complex and TCA cycle enzymes, all in the mitochondrial matrix. Okay, this is a very simple picture. Pyruvate enters into the mitochondria through pyruvate transporter. Why I have shown here this picture is, some of you may get confused whether the pyruvate itself enters into the mitochondria or pyruvate enters after getting converted to acetyl CoA. Don't get confused. Pyruvate has a transporter known as the pyruvate transporter, and this transporter transports pyruvate to the mitochondria. And only after going into the mitochondria, this pyruvate is converted to acetyl-CoA. Okay. Pyruvate enters into the mitochondria through pyruvate transporter. This is phosphoenol pyruvate. We have continued with the glycolysis here for simplicity. Phosphoenol pyruvate is converted to pyruvate. Now this pyruvate is carried inside of the mitochondria with the help of pyruvate transporter, and once inside the mitochondria it is converted to acetyl-CoA. Now only after forming the acetyl-CoA it will combine with the oxalate state to form citrate and continue in the TCA cycle. And in the process of gluconeogenesis, this pyruvate also participates in gluconeogenesis. This pyruvate, with the help of pyruvate decarboxylase, it will give oxaloacetate, and this oxaloacetate with the help of PCK, that is a phosphoenol pyruvate carboxykinase enzyme, it will form phosphoenol pyruvate. This phosphoenol pyruvate has the transporter, and it will go to the cytoplasm, and it will go backward reaction, resulting in the formation of glucose. That is the process of gluconeogenesis is focused here. But the main point is this pyruvate will be carried inside to the mitochondria and it is converted to acetyl-CoA. This pyruvate dehydrogenase is an enzyme, and we say as the pyruvate dehydrogenase complex because it is a complex. How and why it is complex is this is a multi-enzyme complex containing 3 enzymes associated together non-covalently. That means pyruvate dehydrogenase complex is a protein molecule which catalyzes 3 different reactions. That means 3 different activities are present on the same protein. That is, this is the example of multifunctional multifunctional protein or multifunctional enzyme. 3 enzyme activities are pyruvate dehydrogenase, dihydrolipyl transacetylase, and dihydrolipyl dehydrogenase. The name simply is pyruvate dehydrogenase and complex because it is not always easy to remember all the 3 names. That's why we simply call it as pyruvate dehydrogenase complex. And this pyruvate, this is being shown here. Let's try. It's not going. Okay. Pyruvate dehydrogenase complex. By the name itself, the complex is there because this is the multifunctional enzyme. That means the same protein having different enzyme activities, and in this they are having 3 different enzyme activities: pyruvate dehydrogenase, dihydrolipyl transacetylase, dihydrolipyl dehydrogenase. 3 different enzyme activities, and to accomplish these 3 different enzyme activities, it also has the cofactors. Just like this pyruvate dehydrogenase uses thymine pyrophosphate as a cofactor or coenzyme. More correctly, thymine pyrophosphate. This is the coenzyme. Pyruvate dehydrogenase uses thymine pyrophosphate. Dihydrolipyl transacetylase. This is the lipoic acid bound and uses coenzyme A as a substrate. That means as a coenzyme. Next is dihydrolipyl dehydrogenase. This is FAD bound and also uses NAD as a substrate. That means there are 5 coenzymes. One is thymine pyrophosphate, next one is lipoic acid, coenzyme A, FAD and NAD. That means the pyruvate dehydrogenase complex is a multifunctional enzyme with 3 different enzyme activities and 5 different coenzymes or cofactors. Okay, let's see in brief the reaction catalyzed by pyruvate dehydrogenase. This pyruvate dehydrogenase complex serves as a bridge between the glycolysis and the TCA cycle because glycolysis leads to the generation of pyruvate and pyruvate has to be converted to acetyl coenzyme to go to the TCA cycle. This pyruvate dehydrogenase complex reaction serves as a bridge between glycolysis and the TCA cycle. This generates acetyl coenzyme A and NADH from oxidative decarboxylation of pyruvate. You should not forget that in this reaction NADH is also produced. That's why when you calculate the total amount of ATP formed from the oxidation of, for example, 1 molecule of glucose, you should not forget this step because certain ATPs are formed in glycolysis and certain ATPs are formed in the TCA cycle. And when you add that, it will not be the total because some ATPs are formed in this step also. As you can see, it results in the formation of one NADS molecule. One NADS molecule means, according to the old concept, you will calculate as 3 ATP molecules. And when you go for calculation of total amount of ATP formed from one molecule of glucose means there will be two pyruvate molecules formed, and from two pyruvate molecules, two NADS will be formed, and accordingly you have to calculate the MCOMS. Okay, pyruvate will give rise to the formation of acetyl-CoA with the help of enzyme pyruvate dehydrogenase complex. We have already said it contains three different enzyme activities, and for our simplicity we have written pyruvate dehydrogenase complex, and in the bracket E1, E2 and E3. Because it is not always easy to remember all the three names, that's why just to write about the concept, three different enzymes: pyruvate dehydrogenase complex, E1, E2, E3. Okay. Three enzymes and five different cofactors. Yes, what are the five? Okay, you can start from NAD. Second is FAD. Third is coenzyme. Fourth one TPP. Fifth one lipoic acid. These are the five cofactors. Pyruvate dehydrogenase complex catalyzed reaction is irreversible. That means pyruvate will be converted to acetyl coenzyme A, but acetyl coenzyme A cannot be converted to pyruvate by the same reaction. This is irreversible, and this explains why glucose cannot be formed from the acetyl coenzyme A, or simply from the lipid. Why glucose cannot be formed from the lipids? Okay. If somebody asks you question, will glucose be formed from the lipids or not? Generally, there will be no formation of glucose from lipids, because lipids after the breakdown is just like fatty acids, they will form acetyl-CoA, but this acetyl-CoA with a backward reaction, it cannot go to the formation of pyruvate and going backward forming the glucose. That's why lipids cannot give rise to the carbohydrate or the glucose molecules, but there is an exception. Just like in the lipids, you say triacylglyceride. In the three fatty acids, when they are decomposed, they will give rise to acetyl-CoA, large amount of acetyl-CoA. That will not give rise to glucose, but that glycerol molecule can give rise to glucose. That's why if you have to become very specific and particular, very little amount of glucose molecule can be synthesized from the lipids, but simply no synthesis, because only from that glycerol molecule glucose can be synthesized. This explains why glucose can't be formed from the acetyl coenzyme A by gluconeogenesis, because of the irreversible nature. Okay, we are talking about the pyruvate dehydrogenase complex reaction. Pyruvate dehydrogenase complex converts pyruvate to acetyl CoA, and we have simply denoted this as enzyme E1, E2, and E3. And this is the simple equation which we write in the exams, which we all have to know, but we are not showing here the three different enzyme activities and their sequence. Now we'll see them in detail. This okay. Now we have started from here pyruvate, and what is formed is acetyl CoA. How they are formed? Let's see. Pyruvate. That means we have said this enzyme uses five different cofactors. First one is thymine pyrophosphate. This thymine pyrophosphate, first of all, will react with pyruvate, resulting in the formation hydroxyethyl thymine pyrophosphate. In this process, one carbon dioxide will be released. The enzyme, first enzyme is pyruvate dehydrogenase. With the action of pyruvate dehydrogenase, this pyruvate will be converted to hydroxyethyl thymine pyrophosphate by combining with thymine pyrophosphate. This is the first reaction. Now, after the formation of this hydroxyethyl thymine pyrophosphate, now this hydroxyethyl thymine pyrophosphate will react with this oxidized lipomide, and this acetyl group will be donated to the oxidized lipomide with the generation of thymine pyrophosphate. Now this oxidized lipomide will form acetyl lipomide, because this acetyl group will be gained by this, and the enzyme name is dihydrolipide transacetylase. This is the second enzyme activity. Now there is the formation of acetyl lipomide. Now after the formation of this acetyl lipomide, how it reacts is coenzyme A will come, and this coenzyme A will take this acetyl group, resulting in the formation of acetyl coenzyme A. That means acetyl coenzyme A is formed, but there are certain reactions remaining. Now what happens? This is the reduced lipomide Now this will be oxidized. First of all, this is the oxidized lipomide. How it gets oxidized is this NAD takes the electrons, and this NAD gets converted to NADH. First of all, FAD takes the reaction, and this FAD DS2 transfers into NAD, and NAD results in the formation of NADH. At the same time, this oxidized lipomide is generated, and it is available for the next cycle of the reaction. This is the full mechanism of reaction of pyruvate dehydrogenase complex. But you can simply denote pyruvate dehydrogenase complex E1, E2, E3, and results in the formation of acetyl-CoA from pyruvate. And you have to write the name of the cofactors. Cofactors are, if you get easier, if you feel like easier to start from NAD, because this is the one which you usually come across, NAD, then FAD, then coenzyme A, thiamine pyrophosphate, and lipid. These are the 5 cofactors. Okay. There is one clinical application about this pyruvate dehydrogenase complex reaction. Arsenic and organic arsenals are toxic compounds because they bind to sulfhydryl compounds that form bidentate hydrox. What I am going to say is about the mercury poisoning or the arsenic poisoning, isn't it? These arsenic compounds, they bind with the sulfhydryl compounds. Sulfhydryl means this lipomide, isn't it? One part of that pyruvate dehydrogenase complex. That lipomide part, that will be acted upon by arsenite or arsenic. How it is acted upon is this dihydrolipomide is like having the two sides free, but after reactions with this arsenic, now the free sides will be closed. That means now this pyruvate dehydrogenase enzyme will be inactivated, and if pyruvate dehydrogenase enzyme will be inactive, means now there will be no formation of acetyl-CoA from the pyruvate, and there will be less generation of ATP. This is about the arsenic poison. Okay, arsenite and organic arsenals are toxic compounds because they bind to sulfhydryl compounds. What is sulfhydryl compound here? This sulfhydryl compound is pyruvate dehydrogenase complex. Actually, which portion? Dihydrolipomide portion. Inactivation of lipomide containing enzymes by arsenite, especially pyruvate dehydrogenase complex and alpha-ketoglutarate dehydrogenase complex, brings respiration to a halt. Not only this pyruvate dehydrogenase complex, but also another enzyme is alpha-ketoglutarate dehydrogenase complex. This alpha-ketoglutarate dehydrogenase complex we will study in the TCA cycle, but the mechanism of action of both the enzymes is similar. That's why we are also mentioning here in arsenic poisoning. Pyruvate dehydrogenase complex and alpha-ketoglutarate dehydrogenase complex. These arsenic molecules will combine with dihydrolipomide to form the bidentate hydrox, and this is the inactive part. That means it will render the enzyme inactive. Okay, now let's see the regulation. Regulation of pyruvate dehydrogenase activity. How it is regulated? Because after studying about each of the biochemical pathway, we also study about the regulation, because we have to know how they are regulated. Now, regulation of pyruvate dehydrogenase activity. This pyruvate dehydrogenase enzyme is regulated actually by two means. One is the phosphorylation and dephosphorylation, and the other is the direct regulation by the metabolic products. Okay. When you analyze this picture, you can see one is in the red one, and another one is in the green one. And this green one is the active one, red one is the inactive one. The inactive one is attached with the phosphate group. That means the phosphorylated form of this enzyme is inactive, whereas the dephosphorylated form is active. Okay, as you can see, it is phosphorylated, and this phosphate group is removed with the help of pyruvate dehydrogenase phosphatase, and this pyruvate dehydrogenase phosphatase is activated by calcium, magnesium, and insulin. That means in the presence of calcium or in the presence of insulin, this pyruvate dehydrogenase complex will be active and it will perform its work. That means when we are doing vigorous exercises from the muscle, calcium will be released, and at that time, how it is signaled is By the presence of this calcium, now this pyruvate dehydrogenase enzyme will be active. Or in the fasting conditions, so not in the fasting conditions, sorry, in the feeding condition, just like we have taken large amount of milk, insulin will be there, and in such condition also this enzyme will be active. Whereas this pyruvate dehydrogenase kinase will attach the phosphate group to this pyruvate dehydrogenase to form phosphorylated pyruvate dehydrogenase, and once it is phosphorylated, this is inactive. And what are the metabolic products which promote this phosphorylation? They are NADH, acetyl-CoA, and NADH. That means if there is already large amount of acetyl-CoA means this will be phosphorylated and it will be inactive, and there will be no more formation of acetyl-CoA. Or if there is already large amount of NADH also, there is no requirement of this pyruvate dehydrogenase activity or the TCA cycle. That means there is already enough energy. In that condition also this will be inactive. Okay, this is about the regulation of pyruvate dehydrogenase complex activity. Pyruvate dehydrogenase complex is regulated by two means. One is phosphorylation and dephosphorylation. The other one is by the direct regulation by the metabolic products. And this enzyme pyruvate dehydrogenase complex is active in dephosphorylated stage. Okay, now let's see the biological correlation or the biochemical correlation or the biochemical importance of pyruvate dehydrogenase complex. Lack of thymine pyrophosphates inhibit pyruvate dehydrogenase activity, resulting in the accumulation of pyruvate. Thymine pyrophosphate. Okay, any of you tell me, have you earlier heard the word thymine? Have you heard the word thymine?
Vitamin B1.
Okay, B1, yes. That means this thymine pyrophosphate is a coenzyme derived from vitamin B1, or thymine pyrophosphate is the active form of thymine or vitamin B1. Lack of thymine pyrophosphate inhibits pyruvate dehydrogenase activity, resulting in the accumulation of pyruvate, because this cofactor is required for the activity of pyruvate dehydrogenase function. In thymine deficient alcoholics, pyruvate is rapidly converted to lactate, resulting in lactic acidosis. Why we have said about the alcoholics is, in the case of alcoholics, they do not give importance to the cereals like rice after coating of rice, but they give importance to the other foods. That's why they become deficient to the thymine. And in such people, in thymine deficient alcoholics, pyruvate is rapidly converted to lactate, resulting in lactic acidosis. That means if they are deficient in vitamin B1. In such condition, pyruvate dehydrogenase complex cannot function properly because thymine pyrophosphate. Thymine is derived from vitamin B1. In patients with inherited deficiency of pyruvate dehydrogenase, lactic acid is seen after the glucose load. Because in this also, in the same condition, if you give more amount of glucose at the same time, then what happens? Due to the deficiency at the same time, the pyruvate cannot be converted to acetyl-CoA, so it results in lactic acidosis. We have already said pyruvate dehydrogenase complex activity is inhibited by arsenic and mercury ions by the formation of bidentate adducts. We have already seen. This is by binding of these ions with the sulfhydryl groups of lipoic acid, because one part of this pyruvate dehydrogenase was the lipoic acid. Okay, with this we have studied about the connecting link between the glycolysis and the Krebs cycle, that is the pyruvate dehydrogenase complex reaction, and we also correlated that pyruvate dehydrogenase complex with the thymine deficient alcoholics, and also we correlated with the arsenic and mercury poisoning. Now we have already produced the acetyl-CoA, and this acetyl-CoA will go to the TCA cycle. Now we will go to the TCA cycle, also known as citric acid cycle or the TCA cycle. Why this citric acid cycle is known as TCA cycle? What is the full form of TCA?
Tricarboxylic.
Okay. Now it is having the several names, as you can see here: Kreb cycle, citric acid cycle, TCA cycle. Why this cycle is known as the Kreb cycle? Why it is known as the Krebs cycle, why it is known as the citric acid cycle, or why it is known as the TCA cycle. There are three different names, isn't it? Okay, first about the Krebs cycle. Why this cycle is known as Krebs cycle?
Founder's name.
Okay, because Hans Kreb has first described about the TCA cycle. That's why this is known as the Krebs cycle. Why TCA cycle? Tricarboxylic acid, because the first that citric acid and the other two acids which are formed at the beginning of the cycle is the tricarboxylic acids. That's why this is known as the tricarboxylic acid cycle. Citric acid is the first product formed in the cycle. That's why citric acid cycle. Krebs cycle, citric acid cycle, or TCA cycle was proposed in 1937 by Hans Kreb. This is an anaerobic or aerobic. Krebs cycle is aerobic or anaerobic?
Aerobic.
Aerobic. Glycolysis is aerobic or anaerobic?
Anaerobic.
Anaerobic, sure. You might have studied like glycolysis can be aerobic as well as anaerobic, isn't it? Aerobic glycolysis results in the formation of pyruvate, whereas in the case of anaerobic condition, that pyruvate is converted to lactic acid, so that NAD will be regenerated there itself. But in the aerobic condition, that NAD is regenerated in the electron transport chain. Here, Krebs cycle also, this is also the aerobic cycle, but when we study about the cycle, there is no involvement of oxygen. We don't see the involvement of oxygen, but this is the aerobic cycle. Why this is the aerobic cycle is it requires NAD and FAD, and that NAD and FAD are regenerated in the electron transport chain. They can be regenerated only in the aerobic condition. That's why this Krebs cycle is an aerobic cycle. An aerobic cyclic pathway occurring in mitochondria. This is the final common pathway for the oxidation of carbohydrate, lipid, and protein. TCA cycle is the final common pathway for the oxidation of carbohydrate, lipids, and protein. This oxidizes acetyl coenzyme A to produce GTP, which will be converted to ATP, and it also produces NADH, FADH2, which we also calculate as energy. Okay. In this Krebs cycle, acetyl-CoA will be entering, and that acetyl-CoA will be converted to carbon dioxide, and that oxidized state will be regenerated to each cycle. This is amphibolic in nature. Amphibolic means dual. You understand about the dual nature. That means one hand it is anabolic, on the other hand it is catabolic. Okay, if you remember that glucose is broken down into pyruvate to acetyl-CoA, and that acetyl-CoA goes to the TCA cycle, and in that TCA cycle, that acetyl-CoA will be released as carbon dioxide. This is the catabolic perspective we saw. But different intermediates of TCA cycle, they go to different other pathways. That's why different other pathways to form the larger molecules. That's why in this sense, this is anabolic in nature. About its amphibolic nature, we will study in detail later on. Okay. Functions of the Krebs cycle. Krebs cycle provides reduced coenzymes, that is NADH and FADH2, to the electron transport chain for the generation of ATP. It also provides intermediates for other pathways, example gluconeogenesis, fatty acid and cholesterol biosynthesis, and amino acid metabolism. Also reduces coenzymes that are reoxidized through the electron transport chain linked to the formation of ATP. Because in electron transport chain, NAD and FAD are regenerated, and here they are reduced. So that again they go to the electron transport chain for the generation of energy. And this also participate in subtle pathways that subtle reducing equivalence into the mitochondria. Because now we are talking about the electron transport chain that occurs in the mitochondria, but the reactions of this TCA cycle, which is occurring in the mitochondria, are also essential to carry NADH, which is formed in, for example, the glycolysis, to take into the mitochondria, so that from that NADH, ATP will be produced. That means participating pathways that subtle reducing equivalence into the mitochondria. Here the reducing equivalence is NADH, which is produced in the glycolysis. That will be subtle to the mitochondria. Okay. Now these are the steps. That's the steps which all of you certainly will write in the exam. Some of you may not explain about the other perspective, but this about the cycle all of you. And so okay. Here you can see in the blue to note the different steps. In the red, different enzymes are not here. Now let's start. Okay. After pyruvate, acetyl-CoA is formed. This acetyl-CoA will combine with oxaloacetate with the help of enzyme citrate synthase, resulting in the formation of citrate. This is the first step of the TCA cycle catalyzed by citrate synthase. This we have denoted as the entry of the substrate into the TCA cycle. Now, after the formation of citrate, now with the rearrangement reaction, isocitrate will be formed with the help of enzyme aconitase. This isocitrate, with the help of enzyme isocitrate dehydrogenase, it will be converted to alpha ketoglutarate. And in this step, one carbon dioxide is released and also one NADH is produced. That's why this step we have written as first oxidative decarboxylation, because we have entered acetyl-CoA into the TCA cycle. Acetyl means 2 carbon, and those 2 carbon of the acetyl group will be releasing at the carbon dioxide as carbon dioxide. This is the first step. This is the first step in which one carbon dioxide is released. Now this, okay, start from here. Oxaloacetate is a 4 carbon compound. Acetyl group 2 carbon. Oxaloacetate 4 plus 2 6 carbon. This citrate is having the 6 carbon. Isocitrate is having the 6 carbon. But now there is decarboxylation. After decarboxylation, now 5 carbon compound. This alpha ketoglutarate is 5 carbon compound. Again, with the help of alpha ketoglutarate dehydrogenase, there is second decarboxylation. With this second decarboxylation, now this succinyl coenzyme will be produced, and this is 4 carbon compound. This succinyl coenzyme, with the help of enzyme succinyl CoA synthetase, succinate will be formed. In this step, there is the formation of GTP. ATP is adenosine triphosphate, GTP is guanosine triphosphate. These are the energy equivalent. ATP and GTP are having the energy equivalent, and this GTP can easily be converted to ATP. That's why we count this GTP as 1 ATP. Okay, that means here in this step, when the enzymes are reacting with the substrate itself, there is the formation of energy. That's why this is known as the substrate level phosphorylation. That means when reacting with the substrate itself, there is the production of the energy. That's why this is also an example of substrate level phosphorylation. Okay, succinate gives fumarate, fumarate gives malate, malate gives oxaloacetate. Okay, these steps anyway you have to learn it by yourself. Now let's see how many NADH are formed in this cycle. How many NADH can you see here? Now can you see how many NADH are formed here?
3.
Okay, 3.
3.
1, 2. Where is the next? 3. 3 NADH. How many FADH2? How many FADH2?
1.
1 FADH2. And how many ATPs are formed directly? 1 GTP is formed, which is equivalent to ATP. That means if you calculate according to the old concept, that means from 1 NADH, 3 ATP will be produced, and from 1 FADH to 2 ATP will be produced. How many ATPs will be produced in 1 TCA cycle? 1 TCA cycle contributes to how many ATPs?
12.
12.
Okay, because 3 NADH, that means 3 3s are 9, 1 FADH2, 9 plus 2, 11, 1 directly, that is 12. Okay, this is about the TCA cycle. Okay, now let's see the net reaction. If you had read how to write the net reaction, this is also very simple. Acetyl-CoA will come and this acetyl-CoA will be released as to carbon dioxide. That's why simply on the left hand side you will be writing the acetyl-CoA, on the right hand side you will be writing the carbon dioxide. Now you already know that 3 NADH are produced. On the left you will be writing as 3 NAD plus, and you already know 1 FADH2 is produced. On the left it will be writing 1 FAD, and you already know 1 GTP is produced, means, and in the left you will be writing GDP plus PI. Okay. This is how you simply write the net phase. Now about the inhibitors of Krebs cycle, at least one inhibitor you have to know. Okay. Now this aconitase enzyme is inhibited by fluoroacetate. Alpha-ketoglutarate dehydrogenase is inhibited by arsenite. We have already studied in arsenic poisoning. This arsenic also inhibits pyruvate dehydrogenase complex, and that pyruvate dehydrogenase complex and this alpha-ketoglutarate dehydrogenase complex, they are similar in nature. That means they function similarly. This arsenic, it is the inhibitor of alpha-ketoglutarate dehydrogenase as well as pyruvate dehydrogenase. Next one is malonate, is an inhibitor of succinate dehydrogenase. This malonate is the competitive inhibitor. Okay, these are about the inhibitors of the Krebs cycle. Okay, energy produced by the Krebs cycle we have already counted, and here I have counted from the new system, that is NADH giving rise to 2.5 and FADH2 giving rise to 1.5. According to new concept, if we count according to the new concept, it will sum up to give 10, not 12. Because one NADH gives rise to 2.5 instead of 3 and one FADH to 1.5 instead of 2, and when you add it will give sum of 10. That's according to the old concept 12 ATPs will be produced, but now according to the new concept 10 ATPs will be produced. Okay, now let's see the role of vitamins in the Krebs cycle. We have already studied in yesterday's class also what are the coenzymes, and we have also said as B complex vitamins, different B complex vitamins, they act as the coenzyme. Now here let's see vitamins play a key role in Krebs cycle. These are the name of B vitamins. This is the coenzyme contributed by the respective B vitamin, and these are the enzymes. Okay. Alpha-ketoglutarate dehydrogenase. This uses thiamine pyrophosphate, and this thiamine pyrophosphate is derived from thiamine, that is vitamin B1. Okay, succinate dehydrogenase, FADH2 from riboflavin, vitamin B2, and this NADH from niacin, vitamin B3. This NADH is used not only by these, there are others also. I have listed only few here because if we take the examples of alpha-ketoglutarate dehydrogenase or pyruvate dehydrogenase complex, both of them use thiamine pyrophosphate, FADH2, NADH, coenzyme A. All the coenzymes are used by those enzymes, but here also I have listed the names. Okay. About the regulation, okay, we will study in the next class. Up to here, if you have any queries, okay. Just on.
Sir, can you answer the regulation of that pyruvate dehydrogenase?
This is the regulation of pyruvate dehydrogenase. Two types of regulation are there. One is it is regulated by phosphorylation and dephosphorylation, and the next one is by the metabolic products.
Sir, this ATP here, it is decreasing that pyruvate dehydrogenase kinase. It should increase.
Your question, please.
Sir, ATP is inhibiting that enzyme, but it should activate that pyruvate dehydrogenase kinase.
Pyruvate dehydrogenase kinase, actually for the action of this pyruvate dehydrogenase kinase, ATP is required. That means during the activity of this, ATP will be utilized, resulting in the formation of ADP. That means if already there is a large amount of ATP, means this will be active, pyruvate dehydrogenase kinase will be active, and once this pyruvate dehydrogenase kinase will be active, pyruvate dehydrogenase enzyme will be inactive.
Okay, okay.
This not as the along with the plus or the minus sign, but this is to show this pyruvate dehydrogenase kinase requires ATP. Because while studying the glycolysis also, you study like kinases require ATP for their action. That means ATP is involved in the kinase reactions. However, there is an exception in the glycolysis itself, but most of the kinases they require ATP for their action.
Okay, sir. Thank you.
Okay, any more questions?